Hi all, Jean Kirapinha here. In our last video, we delved into the world of decoding research integrity and how to evaluate scientific papers. As a brief recap, what we did, we discussed the immense influx of research papers in today's digital age. And also we discussed the critical need to differentiate credible research from the rest. It's not about doubting everything that we read, but being equipped with the right tools to interpret and assess the integrity of scientific work. Today, we're continuing with this exploration of evidence in medical research, particularly focusing on the traditional hierarchy of evidence structure, RCTs, and we'll just touch upon cohort studies, which will be the subject of the next video. So onto the subject of hierarchy of evidence. In the world of clinical interventions, it's essential to prioritize our decisions based on the weight of different types of primary studies. This systematic prioritization is what we refer to as the hierarchy of evidence. But what constitutes this hierarchy? Positioned right at the top, at the pinnacle, are systematic reviews and meta-analyses. These are secondary research papers where multiple primary studies on a specific topic are thoroughly analyzed and appraised. RCTs come next, with the definitive results holding more weight. These RCTs provide results with confidence intervals that leave absolutely no doubt about their clinical significance. But there are also RCTs with non-definitive results, where the outcome might suggest a significant effect, but the confidence intervals are not as clear-cut, or where the clinical significance is questionable. Following RCTs, just slightly below, are cohort studies, case control studies, and cross-sectional surveys. Each of these offer unique insights and fill specific gaps in our understanding of clinical phenomena. Often seen as the fundamental blocks of medical literature, case reports are at the bottom and are individual accounts of patients detailing the symptoms, diagnosis, treatment, and follow-up. While the hierarchy provides a framework, it's crucial to understand its limitations. Not every well-conducted meta-analysis or RCT will automatically supersede a meticulously designed cohort study. The true value of a study isn't just in its design, but it's also in its execution, rigor, and its relevance. It's also worth noting that many impactful studies, especially in the qualitative research domain, might not fit into this hierarchy that I've just described. Medical science is vast, and understanding its intricacies requires more than just ticking off a study's design against a predefined scale. In our journey today, we're exploring these layers, discern their significance, and understand how they come together to shape the bedrock of evidence-based medicine. By the end of these videos, I hope you would have gained a comprehensive understanding of how to evaluate and interpret the myriad studies that contribute to the ever-evolving field of medical science. So onto a primary topic for conversation today, which is unraveling the science of RCTs. In the intriguing world of medical research, the RCT stands as a stalwart method for rigorous scientific investigation. Imagine stepping into a grand medical theater. On one side, participants receive an intervention, perhaps a new drug or a medical device, whilst on the other side, participants receive a placebo. Both sides are observed, and as the curtain drops, their outcomes such as the HbA1c or blood pressure levels are analyzed and compared. The beauty of this approach and this process is the random assignment. Participants are allocated to each group as if by the toss of a coin, ensuring unbiased results. However, not all is rosy in this theatre of research. Some trials masquerade as RCTs, but lack the crucial element of randomization. These imposters are called other controlled clinical trials, where participants are allocated in a non-random way. Perhaps due to ethical concerns, logistical challenges, or other issues, these trials bypass the golden standard of randomization. Even within the realm of RCTs, there exists this murky middle ground. Some trials employ a quasi-random method, which can unknowingly introduce bias. For instance, if a clinician knows beforehand which treatment a patient will receive, they might subconsciously or not choose participants based on how they expect them to respond to the treatment. There are various flaws, methods in this approach, and lacking randomization that we've seen in the past, and this inadvertently taints these studies' integrity. RCTs offer a precise examination of a specific variable in a defined group of participants. They use a forward-looking design, so kind of prospective data collection. When executed correctly, RCTs can iron out biases, presenting a clean slate for comparisons. They even pave the way for meta-analyses, where results from multiple similar trials can be synthesized. However, it's essential to know that when an RCT is a best tool in one's research, or whether it is the best tool in one's research arsenal, questions like whether a drug outperforms a placebo, or if a new surgical method is superior to an existing practice, or if a dietary change can significantly impact health parameters, are very good and prime candidates for RCTs. This is why RCTs are often wear the crown of being the gold standard. For all of these merits, they are expensive, though. They take a lot of time to implement. This means that multiple vital questions sometimes go unanswered due to the lack of funds or time. Additionally, biases, overt or covert, can sneak in from imprecise randomization and may introduce bias, particularly with the selection of different patient groups. Moreover, the results of an RCT might not always translate seamlessly to the broader population. There's a vast landscape of research questions for which RCTs might not be the right fit. Sometimes when an intervention's success is glaringly obvious, RCTs become redundant. In other scenarios, previous research or ethical concerns might render RCTs completely impractical. So, in the vast arena of medical research, while we venerate the RCT for randomized controlled trials for their precision and power, it's imperative to remember that they are just but one tool amongst many. It's not solely about the design of a study, but the appropriateness of that design for the question at hand. Thank you for tuning in, and until our next exploration, stay curious and remain critical. Take care.